This video will introduce you to the concept of calculating heritability in quantitative traits. So how do we know if a phenotype is tied to genetics or the environment? To answer this question, we need to calculate heritability. Heritability is defined as the proportion of the phenotypic variance that is attributed to genetic variation. Heritability can be calculated in two ways: the broad sense heritability and the narrow sense heritability. The broad sense heritability is calculated as the variance due to genetics divided by the total phenotypic variance. The calculation accounts for all the genetic variants that may affect the phenotype. The genetic variance term V sub G can be further broken down into three components. V sub A is the variance due to additive allele effects. V sub D is the variance due to dominant recessive patterns of inheritance. And V sub I is the variance due to alleles that interact in an epistatic manner. Now, the majority of the time, we simply focus on the V sub A term. And when we only include that term in our calculations, we are actually calculating the narrow sense heritability. To calculate the narrow sense heritability, typically we will measure a quantitative trait among groups of genetically related individuals. This allows us to calculate the correlation between the groups of related individuals and compare that to what we expected the correlation to be. The final value for narrow sense heritability, that is, the value that tells us what proportion of the phenotypic variance is related to genetics, is calculated as the observed correlation, which is what we measured and calculated, divided by the expected correlation if all the variance was explained by genetics, which is based on the known genetic relationships among our groups. Some examples of expected values will be 0.5 for siblings, since they on average share 50% of their DNA. Identical twins share 100% of their DNA, so we would expect a trait controlled by genetics to have a correlation value of 1. We can do this for any set of related pairs of individuals and determine what the narrow sense heritability is for that particular trait. One note here is that there's considerably more calculation involved in calculating the R observed value that we have talked about so far, and that will be the subject of a separate video. To wrap up heritability, it's important to know the limitations of this value. A narrow sense heritability value only says what amount of phenotypic variation is due to genetic variation for a particular population and a particular environment. We can see from this that our value is actually describing a very narrow phenomenon and is restricted to the population we study in the particular environment we studied. This means that in order to understand heritability across broad scales of time and space, we need to explore multiple environments and multiple populations in order to truly account for the relative effects of genetics versus environment. This also stresses the importance of not making too much of these values, something that has unfortunately been done in the past.